everybody. My name is Newton Tran, and I am a master's student in Dr. Arnold Siraki's lab. Today, I will be talking about how the oxidation of Fenamate compounds by neutrophil myeloperoxidase produces toxic reactive metabolites. Starting with some background, Fenamates are a group of non-steroidal anti-inflammatory drugs, or NSAIDs, and are some of the most commonly prescribed drugs in the treatment of pain and inflammation. These drugs undergo oxidation reactions to produce reactive metabolites, and consequently, they exist as a potential toxicological concern due to their abilities to weaken cells and cause damage to proteins and DNA. A potential interaction target for these drugs are peroxidase enzymes, such as myeloperoxidase or MPO. MPO is an antibacterial heme protein that is synthesized in bone marrow promyelocytes and is also found in neutrophils and specific leukemia cell lines. It is known for its strong oxidizing capacity. In this figure, MPO is activated in the presence of hydrogen peroxide to form compound 1, providing it with the ability to oxidize drugs into reactive metabolites. So the main research question of this study is, what is the role of MPO in the toxicity of these drugs? And therefore, we hypothesize that the enzymatic activity of MPO is a requirement for the oxidation of these Fenamate compounds, leading to reactive metabolites, and thus causing toxic effects. In order to do so, our objectives are to characterize the metabolites, determine cellular effects and responses, and understand the basis of metabolism and toxicity. There are 4 clinically used Fenamate compounds: flufenamic acid, mefenamic acid, meclofenamic acid, and tolfenamic acid, all of which are analogs to the parent structure N-phenylanthranilic acid. We started with UV-Vis spectroscopy studies to observe any spectral changes as a sign that oxidation was occurring. Here we see the spectral change from the oxidation of meclofenamic acid at 0 minutes in blue and at 60 minutes in orange, where there is a large peak formation at 450 nanometers, displaying that oxidation is occurring. However, with the addition of glutathione or GSH, this peak disappears. The addition of GSH is intended to observe any formation of GSH adducts with the metabolites, which takes us to our next study, LCMS, to initially assess the metabolites formed from the oxidation of Fenamate compounds. Here we display the chromatograms for the oxidation of tolfenamic acid and its interesting metabolites. From the diode array detection scan, we can see 4 peaks, where each peak corresponds to a different metabolite, and the metabolites being characterized as hydroxytolfenamic acid dimer, and a tolfenamic acid dimer GSH adduct. These peak characterizations are based on the mass to charge ratio and retention times, and only the proposed structures are shown. Moving on to cell viability studies to assess the cytotoxicity, these drugs were seeded in a dose-dependent manner in human leukemia HL60 cell lines, which are abundant with MPO. We found that meclofenamic acid was the most toxic, and N-phenylanthranilic acid was the least toxic based on their IC50 values. We then looked at electron paramagnetic resonance studies, or EPR, to assess the formation of any glutathionyl radicals. The reactive metabolites oxidize GSH, forming a GS radical, which can be trapped using a spin trap reagent such as DMPO. In this figure, we see that all Fenamate compounds produces GS radicals in a linear dose-dependent manner at different rates, with meclofenamic acid having the highest signal response and flufenamic acid having the lowest signal response. In conclusion, MPO was observed to catalyze the oxidation of most drugs except for N-phenylanthranilic acid. From LCMS, hydroxyquinonamine and dimer products were observed for most Fenamate compounds, although GSH adducts were only observed for N-phenylanthranilic acid, mefenamic acid, and tolfenamic acid. And lastly, meclofenamic acid produced the most glutathionyl radicals and was observed to be the most toxic Fenamate compound.